All right, so on Monday the 14th, we talked about various scientists. We talked about how early in the 20th century, scientists didn't know for sure what molecule was responsible for the storage and transmission of genetic information from one generation to the next. They were looking at proteins being the more likely molecule, maybe DNA, but they were leaning towards proteins because there are 20 different amino acids, which would give more variety. DNA, there's only 4 different nucleotides. We discussed Frederick Griffith and his experiments with the R strain and the S strain pneumococcal bacterial cells, and how he determined that heat can kill the super deadly S strain, but when mixed, when heat killed and then mixed with live R strain, it still resulted in the death of the mouse. And then we talked about Oswald Avery and his colleagues and how they extended on that and how they took that, that Griffith experiment one step further and had the addition of different enzymes to target certain molecules. So they used protease, which destroyed proteins. They used RNAs, which destroyed RNA, and then DNAs, which destroyed DNA. And they still repeated that last part of the Griffith experiment where they heat killed the S strain, treated it with protease to kill the proteins, then mixed it with the live R strain. And the point was to destroy the proteins to see if it was still going to cause that transforming principle that Griffith had discussed after his experiments. And when the heat killed S strain was mixed with the protease, the mouse still died. Same thing with the heat killed S strain with the RNAs, mixed with the live R strain, mouse still died. But when they used the DNAs, the mouse still lived. So they were pointing to DNA being the likely substance that was transformed from the dead S strain to the live R strain. But of course, in true scientific fashion, a lot of scientists weren't convinced. They started trying to poke holes in it. They said, well, maybe it was contaminated by a protein that couldn't be denatured by heat or protease. But a lot of people still started to say, well, maybe there's something about this DNA. And I want to stress that it's not a bad thing that scientists were saying, hey, Oswald Avery, maybe your samples were contaminated, because that's scientific process. That's really investigating it further. I mentioned this in class on Monday. It's not one experiment that is the end all be all. You need lots of evidence to support claims, to support hypotheses, to allow them to become well-supported theories. So then Alfred Hershey and Martha Chase in 1952 used bacteriophages and some radioactive phosphorus and some radioactive sulfur to see what was transmitted by the bacteriophage to the cells that they infected. And they determined that it had to be the DNA, because the only ones, when they looked at the offspring bacteriophage that were replicated by the bacterial cells, they couldn't find the radioactive sulfur. So that means the radioactive sulfur was not used to create the next generation. It was not transmitted. Whereas the radioactive phosphorus was. And if you remember back on Monday, we talked about the one element, the element difference between proteins and DNA was DNA contains phosphorus and proteins contain sulfur. So if it was proteins that were transmitted, then we would have found that radioactive sulfur in the new generation of bacteriophages, but we didn't. And if you missed those notes, they are posted up on Blackboard under the in-class notes module, and you'll see the date in class notes 14 October. But let's get into this last page that we didn't get to on Monday. So I'm going to actually type into this because it's just going to make life easier, and I want, maybe I can make it look all fancy. I was trying to see if I had a font size, but I guess not. All right, so what in the world? Why are you not? Technical issues, so sorry. Scientists knew that DNA was made of nucleotides. And normally, and I've said this in class, I prefer handwritten over typing, but I'm trying to save time and type. I don't know if I want that green. I guess I'll just do. I was trying to make it stand out. I kind of like the green. So they were made of nucleotides, but we didn't know what DNA looked like. We didn't know the structure. So in 1950, the scientist named Erwin Chargaff, he made an observation, and we haven't gotten into the type of nitrogen bases, and that's going to be the next video I'm going to post about the structure of DNA. But there are different types. There's purine and there's pyrimidine, and it's based on the ring structure. But what he found, I can't spell. So I might kind of let this hang off the page a little bit because it's a lot. So he discovered that the number of adenine, should it let me? Ooh, maybe. It's just going to make all of it. All right. I'll just go under and underline or highlight, or hey, adenine equals the number of thymine. Cytosine equals the number of guanine. So I'm going to kind of write off here. So A equals T and C equals to G. And that's, we could reverse that. G equals C, C equals A. This becomes important because it lays the foundation for figuring out our base pairing. And we haven't gotten into DNA replication, but base pairing is important because DNA has a double helical structure. So there's one side here, and they have to match. So he's laying the foundation for the next couple of scientists that we're going to talk about. So we have Rosalind Franklin over here in this image. Maurice Wilkins is not pictured. We'll get to them in a minute. We'll actually get down here. So Wilkins and Franklin actually ended up working together at King's College. This was in London. Franklin was hired after Wilkins, and I'm actually going to link up a video to a TED Ed video talking about them. Wilkins assumed he was in charge, and Franklin was his assistant. Franklin kind of was like, no, I was hired to do my thing. But she used X-ray crystallography. and she took pictures, and her famous picture is photo 51, and you can see a faint, it kind of ends up looking, let me get my pen back. It kind of ends up looking like this this kind of sort of X that kind of goes like this. I'm kind of sketching it out to the side here, and it's my recreation. And if you don't know what you're looking at, you don't really know what's going on. But for this particular one, they realized right away that that pattern has to be a double helix. So let's kind of summarize that. So Franklin used X-ray crystallography. Oh, there's supposed to be a C there. To determine that DNA was most likely a helix. So later, Wilkins showed her data to Watson and Crick, who weren't really too concerned about her at the time. But interestingly enough, I'm trying to scroll up here. Here we go. First time using the whiteboard in office. So we have James Watson and Francis Crick. They worked also in England, but they were in Cambridge, and if I remember correctly, maybe 50 to 100 miles apart. So close-ish. But also, I mean, they have better transport there, so trains back and forth. So they were working on this too. They were kind of competing with other scientists to try to figure this out. But they used that data that Wilkins gave to them from Franklin's lab and they worked out the accurate model, the double helix structure. The thing is that they did publish their findings in 1953, but they were published at the same time that Franklin and Wilkins published their information, but it was kind of like an afterthought. The way it was published is it was kind of put later in the issue, so it looked like Watson and Crick came up with it, and that Wilkins and Franklin kind of confirmed it. But it's really Rosalind Franklin's work that enabled this. I'm checking my time here. So let's summarize that. Used data given by Wilkins. Franklin's data to come up with model of DNA structure. Interestingly enough, so Watson, Wilkins, and Crick won Nobel Prize for their discovery. Checking my time because it's got me on a time limit. In 1958. But unfortunately, Rosalind Franklin had passed away in 1958, and they don't give you a Nobel Prize after you die. So they won, but she's the one that really helped their case. I think I'm out of time.